Appreciate it.
Gorgeous. Put it right here?
Put it here.
Okay then, the floor is yours.
Great.
Thank you very much.
Am I on? Am I on now? I think so. You hear me? Okay, good. Well, thanks for a wonderful introduction. A pleasure and honor to come here and tell you about some of the work we've been doing at Northwestern. This has been a really exciting couple of days. I'm still a little bit jet lagged. I'm absolutely amazed that we have this size audience after the taps have been open for a while and people are very full from a nice meal. So I guess I have a big challenge to keep you entertained and hopefully get done, as somebody said, in time for the party that's soon to follow. I also have to say I was really impressed by the students. That was just a spectacular demonstration. I didn't meet all of them, but I do want to throw a shout out to Olga and Eric, who got me a lot of airtime in the video, which is very important, and spent really a great deal of time trying to figure this stuff out, and I thought just did a spectacular job, all of them, in terms of distilling really difficult questions down to some reasonable answers and some exciting answers. Also want to thank Roland and Bert for inviting me here. Over the next 45 minutes or so, I'm going to try to tell you about some of the work that we've been doing at Northwestern that I'm extremely excited about. And it fits in nicely with some of the themes of some of the questions that were asked. And in particular, how do you use inspiration from biology or Mother Nature to begin to develop new materials that can have an impact in a variety of areas. First of all, in the construction of new materials. Second of all, in the development of powerful new tools, analytical tools that can impact two, I think, very important areas. One is molecular diagnostics, the development of tools for sensing in the medical arena, and then most recently, the development of a very powerful gene regulation platform. And as I think students emphasized very appropriately, everything that I'm going to talk about starts with fundamental research and discovery for the sake of discovery, trying to understand how things work, how you can make things that haven't been made before, looking at new properties, and then asking how you can use those properties to develop new types of applications. I have kind of a unique position at Northwestern, a very large effort where I can put lots of different people together to take things from beginning to end. And I like to do that because I like to see everything ultimately transition that we do to something that can either impact people in terms of learning or impact people in terms of creating new tools that drive their research forward. And hopefully you'll agree some of this is interesting. So I thought what I'd do is I'd focus on some of the materials and tools that we've developed and specifically nanotechnology-based ones that can impact medicine and, as I said, biology. And so down here in cartoon fashion and picture fashion, we have a whole series of different types of constructs and tools that have been developed. A lot of people talk about the age of nanotechnology coming. You know, when are we going to see some major advances that ultimately hit prime time? There already are examples, and in fact, I'm going to tell you about one, a diagnostic tool that we've developed based upon some nanoparticle chemistry that was really discovered in the late 90s and then transitioned over the last decade into a very powerful diagnostic system that now is used in hospitals all over the world, including here in Europe, and will, I think, soon be used by almost every hospital, because what it allows you to do is to create very high sensitivity and high selectivity diagnostic tools that open up the field of point-of-care medicine. And so I want to introduce you to some of those and talk about some of the materials advances that really led to the development of those types of tools. And so these aren't, again, pie-in-the-sky type systems. These are FDA-cleared systems, so pretty remarkable, I think, at least when you think about nanotechnology, the modern age really starting roughly a decade and a half ago, and we now have such tools at our disposal. If you think about where nano can play a role in these areas, I've just highlighted a few here. So high sensitivity and high selectivity molecular diagnostics, in vivo imaging materials, better contrast and theranostic capabilities, so combined therapy and diagnostic capabilities, tools for genetically manipulating cells and making intracellular measurements. I'll tell you about some really neat types of particles that allow you to get entities into cells and to go in and actually make measurements in live cells so that you can begin to differentiate cells based upon their internal cargo for the first time when we're talking about looking at genetic material, for example. The old way is to grind them up and measure the genetic content. But opening up the ability to do live cell imaging would be a big deal, and we'll talk about why that is in a little bit. And then I want to finish with some new and potentially better approaches to drug delivery and gene therapy. And I really want to focus on not just a different way of doing things, but why nanomaterials offer a much better way of doing things in certain cases. Rather than telling you about all of those constructs, I want to talk about my favorite one. And I now refer to this type of structure as a spherical nucleic acid. That's kind of an interesting term. In fact, if you search it on Google, you won't find it. We coined it about a month ago as I was writing a review for JAX. Because I thought about what we were doing, and effectively over the last 15 years or so, we've learned how to take particles and functionalize them with oligonucleotides. And when you do so under the right types of conditions, you can begin to make spherical architectures, three-dimensional architectures, that have oligonucleotides that are highly oriented and densely packed. Those types of structures have fantastic properties. In addition to taking on some of the molecular recognition properties of the oligonucleotides immobilized on their surfaces, they have a whole series of cooperative properties, properties that derive from, as I said, the orientation and dense packing of the oligonucleotides and the local salt environment that's created by doing this. Structures that have, as I said, properties that you could have never predicted prior to making these types of systems. And we weren't thinking about developing diagnostic tools or gene regulation materials when we made these structures. We were simply thinking about beginning to make a construct that would have programmable recognition properties by virtue of the oligonucleotides put on the surface of the particles. And then the idea was we might be able to use oligonucleotides to assemble those particles, like artificial atoms, into extended materials. So materials by design, building from the bottom up, structures where we could control the placement of the particles in 3 dimensions, in the periodicity, with tremendous precision, at least in our mind, back in the mid-90s. This is really the story here. So for those of you that had too much beer, just wake up a little bit, look at this, and remember this, because I think it's actually very important. When you look at these types of structures, spherical nucleic acids, and compare them to their linear counterparts, they are radically different. And that's when the sequences are identical. So the only difference here is that we have a lot of oligonucleotides oriented on the surface of a particle versus oligonucleotides free in solution. If you look at, for example, when they bind to complementary nucleic acids and their melting properties, over here what you find is you get very narrow melting transitions. They exhibit what's called cooperative melting. And we've spent a lot of time studying those and figuring that out. I'll talk to those that are interested afterwards about why that is. But take my word for it right now, these types of transitions occur sometimes over a single degree. I have 2 to 8 degrees here. That's a typical type of range. Over here we have 20 degrees. Why is that important? Well, if you use these particles as probes, what that means is you can differentiate perfectly complementary targets, for example, in a detection system, much more easily than you can, with a higher degree of fidelity than you can with the free oligonucleotide type of probes. For example, the fluorophores on there. So I can begin to get higher selectivity diagnostic tools when I use these as nanoparticle probes. They bind complementary nucleic acids about 100 times better on a sequence-for-sequence basis. Again, a lot like chelation and inorganic coordination chemistry, but you have a local environment that's very rich in salt that stabilizes duplex DNA. Why is that important? That means I can fish out a lower concentration of oligonucleotide targets in solution. That turns out to be important in both diagnostics and, as I'm going to show you, intracellular gene regulation. This has a core that has tailorable physical properties and chemical properties for that matter. So we can tailor the plasmonic, the catalytic, the magnetic, and the luminescent properties. This, of course, does not. This is amazing. Cells, we talked about artificial cells. Cells have designed ways of keeping out foreign nucleic acids. So free oligonucleotides will not naturally pass cell membranes. These guys go through better than the known agents designed to carry in nucleic acids, polymer-based agents, and we'll talk about why that is. So again, completely different behavior based upon structure here. Immune response. These trigger almost no immune response with respect to cells. These have very elevated immune responses, and you can measure that based upon interferon beta levels. And then this is the other one that's very important on the regulation side of things. Inside cells, cells also have machinery, nucleases designed to chew up foreign nucleic acids. These have a very short half-life, as they should. These actually have a very long half-life. And so the structure actually protects the oligonucleotides from destruction, and you then have longer therapeutic life if you're thinking about this in the context of therapies. Okay, how do you make these types of structures? Well, this was the chemistry we've developed now over the last decade, or 15 years, where we've taken nanoparticles that are typically citrate stabilized. That's important. That's a loose coordination sphere. We take oligonucleotides, typically that have alkyl, thiol, or disulfide end groups. You can't introduce these quickly. I always say that DNA and many of these nanoparticle compositions or colloids are naturally incompatible because salt often destabilizes a colloid and causes it to crash out. So what we do is we slowly introduce the oligonucleotides. The sulfur groups bind to the surface of the gold, and in fact the oligonucleotides lay down on the surface of these particles in the early stages of functionalization. And now we slowly add salt, sodium chloride for example. That weakens the electrostatic interaction between the oligonucleotides on the surface and also the oligonucleotides and the particle surface, allowing you to pack more oligonucleotides onto the surface of the particle. At the end of the day, if you have a 30 nanometer particle, just for benchmark point of reference, we've worked this out for all different sizes in the 5 to 200 nanometer range, you have about 200 strands per 30 nanometer particle. This now is an incredible particle. It is rock stable. You can put it in brine, it just stares at it, it will not aggregate, unless you add complementary oligonucleotides. So you can begin to design particles, for example, like the one shown here. I call this A and this one B, it's just different sequences. If I add a linker now, and again, keep in mind in the early days we were thinking about building materials using this particular approach, that's why we call this a linker, the particles would assemble into an aggregate structure. The reason we began to move towards diagnostics is because we made this observation. You could actually follow this assembly event by looking at this beautiful red to blue color change. And the reason that occurred is as the particles get closer together, they have a plasmon resonance, and those plasmons can begin to interact. That causes a redshifting of the plasmon resonance, which gives rise to this spectacular red to blue color change. When you calculate the concentrations at which this experiment is carried out, what you find is that you're actually amplifying the response associated with that recognition event. You couldn't see this, for example, by UV vis without the gold particles there. We're working at very low concentrations. It's in the low to mid picomolar concentration regime, which is impossible to follow by UV vis spectroscopy just by looking at the native DNA signatures. So immediately we thought what we have is a very simple colorimetric detector for nucleic acids. So we could actually think about these types of particles as probes, and this is a target, and now we could detect this in a very simple format, a lot like a pH test for nucleic acids. Then we started looking at the melting properties of these structures. And you see the second property that, again, we would have never predicted, but we now understand. You see this incredibly cooperative transition. In fact, if you look at it on a reverse phase silica gel plate, it's blue at 58, it's purple at 58.5, and it's red at 59. A transition which normally here in black spans 26 degrees is compressed down to a single degree. And we already talked about why that's important. So again, beginning to look at unexpected fundamental properties and then thinking about how we can ultimately use those in terms of detection. Now I did a bit of a bait and switch. Remember I said our dream was we wanted to learn how to take DNA and use these particles as artificial atoms and assemble them into these three-dimensional networks where we could control with incredible precision the placement of different particles within that three-dimensional lattice. Well, this is not a three-dimensional lattice or at least worth writing home to mom about. It actually is just a polymeric structure. It's amorphous. It has no real order. Well, I'm going to jump forward a little bit because we spent a lot of time, and I'm going to tell you about some of the diagnostic stuff. I'm going to come back to it in a couple of slides. But we came back to this idea after being derailed a little bit on the diagnostic side to think about how we could literally program the formation of crystal lattices. And some students found the trick to do that. In particular, a woman named Abigail Jean-Luton, who's now working with Bob Langer up at MIT, they figured out that if you make really short links that can be involved in recognition and assembly, you can have a system that's dynamic enough that if you've designed it properly, it will fall into a thermodynamic sink, which is in fact a crystal lattice. And we've created a whole series of design rules that allow you to make different types of structures, but the primary design rule is the structure that will be favored is the one that maximizes hybridization. And so this really simplifies the whole concept of bottom-up particle assembly to understanding which types of structures will maximize the degrees of hybridization in the assembly process. So take, for example, two simple structures, FCC and BCC unit cells. I can take one batch of particles, for example, and if I want to form an FCC structure, I add a linker that can hybridize here, and it has a sticky end. I just put it in green, that's self-complementary. Why is that important? Well, what that means is that every particle that comes in contact with another particle can engage in hybridization. They all recognize each other. And that means the structure that will be favored is the FCC structure because that maximizes the number of nearest neighbor contacts. And in fact, if you take this system and anneal it, it falls right into a beautiful crystal lattice. You can tell that by going to the synchrotron and looking at the small angle scattering, and you get a beautiful FCC pattern. What about the Bcc structure? To get that, we simply take this and separate it into two batches. To one batch, we add a strand that has a blue overhang. Important point here is blue does not recognize blue. It's not self-complementary. The other batch, we add a strand that has a red overhang. Red does not recognize red, but these have been designed so that red does recognize blue, and now you have an AB type structure. And so the thermodynamically preferred structure should be the Bcc unit cell. And in fact, that's exactly what happens. And so, we published this, and it was kind of a tour de force type of, at least for us, initial demonstration, because that was always the challenge that I throw out to unsuspecting graduate students. I want you to show that we can take one batch of particles, and by sprinkling in one type of DNA, form one structure, and by sprinkling in another type of DNA, form a different structure. And so this was the first, admittedly the easiest one to do, because you can kind of rationalize it. Well, this has come a long way. We now know how to not only make these types of structures, but once you have this structure, the neat thing about this type of lattice engineering or materials engineering is once I have the sequences that work, I can systematically change either the size of the particles over a fairly large size range, or the length of the DNA, but keeping the recognition element the same. And if I do that, I will always either get the FCC or BCC structure, and I can change the lattice constants in this case. And I can do so with tremendous precision. And we've now learned how to embed these in resins where we can even get electron microscopy images of the resulting structures. And if you carry this a little bit further, what you find is you can now begin to piece together a whole series of strategies to make almost every type of structure known, and some structures which have no mineral equivalent nature, including the ones shown here, right here. So if you look at this and you say, I'm not going to go through all these. If you're interested, there's a paper that was just published in Science that kind of lays out all of these design rules for making these structures. But you might look at this and say, how do you go to something like cesium chloride? Well, one way to do that is you say, I need to go to two different particle types. Make the BCC structure, but go to two different particle types, right? So that I have, let's say, small and big. The problem with that is when you do that, now you have packing parameter problems, right? Because I can put many more small particles around the big particles. But one of the tricks that we employ here is we can take the small particle and make it effectively as large as the big particle by increasing the length of the DNA, but keeping the recognition element the same. So we can change the hydrodynamic radius of that structure so that it'll pack as if it were forming the BCC lattice, but of course gives us the cesium chloride lattice. And it turns out you can play a lot of tricks to get all of these different types of structural types. And I think this becomes a very powerful way of realizing what we set out to do, which was to learn how to literally create design rules that allow you to program from the bottom up different types of lattices where we can control with sub-nanometer precision. That's how predictable this is. Sub-nanometer precision, the placement of the particles in the context of these types of structures. Okay, let's go back to these properties. So we're going through these, and we've talked about two of them. They have fantastic scattering properties as well. So one of the things we discovered was that you could create chip-based assays where you could spot DNA on a chip, a typical microarray, for example, capture a target, and then use the nanoparticles as probes so that when they hybridize to that target, and if you use, for example, a glass microscope slide as a waveguide, you could bring light through it and measure the scattered light coming off of it. You could begin to create colorimetric probes based upon the size and composition of those particle labels and take advantage of those sharp melting profiles to get very high selectivity assays. Then we figured out you can actually use the chemistry to do a trick here to really enhance the signal associated with this. And in fact, this is the basis of that commercial system that I was telling you. This was published in 2000 in Science. What we found is that you get one level of signal if you just use the particles as probes themselves. But if you take advantage of the fact that gold will catalyze the reduction of silver 1 in the presence of hydroquinone, you can flow photographic developing solution over this chip, and anywhere where you have particles present, you plate out silver, and you can increase the signal by a factor of 10 to the 5th in less than 5 minutes. Now you have a system that's extraordinarily sensitive. And as I said, that's the basis for this commercial system. And I'm not showing this to kind of pawn off the commercial system. We actually now use it. Some people said, why do you do the companies? Besides, the obvious of trying to get the technology out there and maybe getting rich from doing it. Well, the other reason is we get all the prototypes and they come back into our lab. And this turns out to be the highest sensitivity diagnostic system out there that does not require the use of enzymes like polymerase for PCR-based type reactions. And in fact, that's one of the beauties of it. It eliminates the need to use those types of technologies, and that opens up the ability to do a variety of things. One is multiplexed assays, where you can simultaneously look at many targets. I'm going to show you thousands in a second. And you can do it very, very high sensitivity. Not as sensitive as PCR. Actually, PCR is a little bit more sensitive. The beauty of this is this eliminates the need to use PCR. It's very difficult to do multiplexed PCR-based assays. And of course, it's remarkably simple. And that's led to a whole series of FDA-cleared assays that are now on the market. Okay, so here's an example. The way these assays look is, let's say we have a chip here. We're looking at 4 different DNA sequences designed to capture hepatitis B, smallpox, Ebola virus, or HIV. A lot of nasty targets. Fortunately, we're only using the nucleic acid, so we don't have to work with the bugs in this case. Anywhere where we have a captured target, we get a developed silver spot. You're looking at a false color image of this, where we're bringing light through and we're measuring the scattered light. And this is designed so that orange is high in terms of light scatter, and this is low. And so there's a quantitative aspect to this. The instrument actually quantifies, not only identifies, but it quantifies the presence of targets. And so then you can look at this, you can read the chip out. You can say this has only got 1 hepatitis B, smallpox, Ebola virus, and HIV. And then this has got 2, 2, 2, 3, and 3. And you see typically what you should see, which is background with these. You don't have perfect differentiation in this case, but the instrument then can differentiate the signals and set a threshold and tell you whether you have a real positive, and if you have a positive, how much target you actually have present. Now, why is this interesting? So this is kind of neat. This is unpublished work here. Recently, we figured out that there are a whole series of new exciting targets that are coming out in the biological literature, and they're called microRNAs. And they float around even in the blood, but they're at very low concentration. In addition, they're also very short oligonucleotides, very short targets. That's not good for doing things like PCR. In fact, it's very difficult to do PCR on these types of targets. So when we saw this, we said this is actually perfect. What we might have is the ability to go through and sort out these types of targets in, for example, blood, and then be able to profile them and identify them and correlate them with disease. And so this chip, which is an in vitrogen chip designed to capture these types of targets, just shows you the power of this. These are all developed spots, silver, measuring the scattered light coming off them. What's interesting about this, the comparison then would be, and I'm a big person in terms of benchmarks. I always tell students when they say, we've got something that's really good, well, tell me good compared to what? Well, the only other way I could think about doing this would be, other than really long, complicated processes like deep sequencing, would be to use a fluorophore-labeled oligonucleotide and develop it by fluorescence. If you do that, you miss 85% of the microRNAs that are present. So what this extraordinary sensitivity does from all of these fundamental properties gives you the ability to see things that you would ordinarily miss in the conventional types of experiments. And I think that's one of the real powerful capabilities that's afforded by at least this type of nanotechnological advance. What about other areas? Well, you could say, well, that's nice, you're detecting nucleic acids, but we have things like PCR, and yeah, it might be better, it might be faster, it might allow you to do a lot of targets, but there are other ways of doing it. And so we like to think about what you can't do. Wouldn't it be great if we had a way of detecting really low concentrations of proteins? PCR revolutionized the way we study biological systems, the way we develop diagnostic tools, because it allowed you to fish out very small numbers of targets. We don't have that on the protein side of things. So we began to think about that, and to accomplish that, we came up with something called the Bio Barcode assay, and it built off of this conjugate again. The basic idea here was, let's say you wanted to detect a protein. We can't amplify it by duplicating it like we can in PCR, and I haven't told you a way to really amplify it with the nanoparticle probes yet. The idea was you'd have to be able to capture a really small concentration of that target in solution. So what we could do is we could add a magnetic particle probe that's just a big polystyrene bead with a magnetic core with an antibody that can recognize the target and capture it. We could then design a nanoparticle probe that has many strands of DNA, exactly what I've shown you today, but also has attached to it antibodies at the terminus that can bind to the captured target. And so if I add a high concentration of that, I can create sandwich structures like this. The gold alone is not magnetic, but when you form the sandwich structure, it becomes magnetic. And so now I can add apply a bar magnet, pull that to the side of the reaction vessel, get rid of the rest of the material in solution, all the excess particle probes, for example. And now what I can do is I can chemically dissolve the gold particle simply by adding cyanide, for example, or iodine, and I can release all of the strands of DNA. And we call those barcode strands. They have no significance in terms of recognizing target. They have no natural significance. What they are addresses and surrogates for the target that we recognize, because we've just traded now one particle for hundreds to thousands, depending upon the size of the gold particle we use, strands of DNA. And now, this instrument that I just showed you is set up beautifully. This is an example of coming back into the lab to sort those barcodes, identify them, measure the concentration, and back calculate the starting concentration of the protein present. What's that going to do for you? Well, I think it's going to do a lot. So if we look at this, the state of detection right now, when you go get a protein-based test, for example, in a molecular diagnostic lab, they're using ELISA technology. That works in the picomolar or higher concentration regime. The barcode assay actually allows you to work up here, but it also allows you to begin to get down into this part of the analytical scale. And I think that's going to be field-defining in terms of capabilities. Why? I've changed the radar. I can begin to look at markers that I missed with the conventional diagnostic tools. I might even be able to study things in individual cells as we get better and better at this. Let me give you one example of where I think that's going to be important. So I don't think you have to think very long. Being able to track markers at really, really low concentration is going to be important in Alzheimer's disease, where the concentrations might be high in the brain but low in the blood. Cancer, where residual disease is critical. A little bit of cancer is a bad thing. Mad cow, you pick pulmonary disease, cardiovascular disease. In fact, there are a whole series of commercial tests that are being developed based upon this, where they can track the early signs of a heart attack based upon the protein called troponin I that's released in your bloodstream as you begin to have a heart attack. And those concentrations elevate in the bloodstream. Well, you have to wait till you get to a certain level with conventional technology. This allows you to track it at much earlier states, and we hope make a difference in terms of diagnosing that disease earlier. But let me give you one where it's clearly going to make a difference. And so I had a urologist in my group. So somebody said, in the introduction, Roland said, professor of medicine. The great thing about that is I can take medical students occasionally. And this guy calls me, his name is Shad Fax, and he says, I want to work in your group. And I said, I have no use for a doctor in my group. I've never done anything in this area. I don't even know what I'd do with you. And he said, well, what if I'm free? And I said, okay, well, now you're speaking my language. I said, are you free? He said, no, I'm not free. I said, well, how are we going to work this out? He said, well, how about I go get a Howard Hughes postdoctoral or a predoctoral fellowship? If I do that, I can join your lab. And I said, you sure? Yeah, I'll keep him busy. Go do that. Well, he calls me 7 months later. He says, okay, I've got my Howard Hughes. I'm ready to start. So I said, get in here. So he starts in. He starts working with the biologists and some of the chemists. And he sees this assay. And he says, you know where this would be really good? His name is Shad Faxton. He's now a professor, actually, at Northwestern. He said, this would be fantastic for looking at PSA. And I go, well, you don't want to look at PSA for this type of test, because everybody knows that all the men in this audience that have their prostates have a pretty high level of PSA in them. You can use standard ELISA technology to measure PSA values. And in fact, that's what's used to track whether or not you have a problem. A lot of people argue about whether it's a good marker or not, but that's what they use. So high sensitivity is not the sweet spot for PSA. He said, no, you don't understand. He said, I'm a urologist. He goes, when you take out somebody's prostate, their PSA levels drop to 0. And I said, well, they're probably not 0, are they? And he said, no, they're not. He said, I don't think they are, but nobody knows. He said, in fact, they're assigned 0 because that's just not detectable. So the question became, could we begin to use this type of test to, first of all, prove people have PSA levels post-prostatectomy? And then, could we begin to use it to track the disease? And here's the neat thing. A lot of people argue about whether a PSA is a good marker or not for prostate cancer. Nobody in the medical community argues about whether it's a good marker for recurrence. If your PSA level rises post-prostatectomy, you are recurring from this disease, and it's a bad disease at that point. So we began to look at this, and we actually finished a clinical study. This was published in PNAS recently. And you can see I just took a couple of snapshots here just to give you a feel for the power of this. So in blue, we have data from the barcode assay. I have up here the limit of detection of ELISA is up way off scale. This is 30 picograms per mil. We have to get up to 100 before we can even begin to detect by ELISA. So all of these guys register, or all these data points are zeros by ELISA. This person's in good shape because all these are post-prostatectomy too. He's flatlining. He's not going to recur. He's cured from that disease. This guy initially thinks he's in pretty good shape. In fact, all these measure as zeros, but he actually recurs. This begins to rise, and you get curves like the one shown here where they rise, and eventually you can begin to use ELISA and track these. So what does that do? Well, I can take now these
patient population post-prostatectomy and use this ultra-high sensitivity assay, and I can say, okay, you're part of the 58% that's cured, you flatlined, you're in great shape. Weight of the world's off their shoulders. They don't have to wait 7 years to get, quote, zeros to know that they're actually cured. These guys are not in such great shape because they're going to recur, and the bad news here is that there is no known therapeutic to treat this disease at this point. But the good news from a research standpoint, and using this type of tool, is I can't create a therapeutic unless I have a way of monitoring whether or not the person's responding to the therapeutic. And so we're now beginning to use the fact that we can get these snapshots of PSA levels and look at whether the person's responding positively, i.e. PSA is flatlining or dropping as a function of therapy or not. And I think this is going to lead to new therapeutic tools because of this type of capability as well. And this is just one example. I think, the interesting thing about this, when I started talking to the medical community, they go, oh, we don't need higher sensitivity. We're having a hard enough time dealing with ELISA and what we do with all that. It's incredible how fixated people get on these types of things. But when you begin to challenge them, I say, look, I'm not telling you this is the way it is. I just want you to think about it. It's amazing. 2, 3 weeks, a month later, they call you and say, I've thought of a million ways of using this type of tool to begin to track new types of markers. And so what we're trying to do now is to begin to use it to begin to discover ways of tracking disease in very different ways because of the high sensitivity. Okay, the last thing I want to talk about now is this idea of therapeutics. So we've talked about materials design, we've talked about diagnostic tools. Can we begin to make an impact in this area of nucleic acid therapeutics? This is really powerful, right? The idea, won a Nobel Prize already, but it's far from realized in terms of potential. The idea that you can go into cells and flip genetic switches and downregulate or upregulate the expression of genes. Very powerful from a research standpoint, not so powerful from a real therapeutic standpoint. In fact, very far in most cases. There are a couple of examples, but very far in terms of really realizing the promise of it for a variety of reasons. I'm going to show you a couple of examples. We've got many, but this will simplify it for the non-aficionados. There are two basic pathways that we can look at. One is called antisense, and the other is called siRNA. In antisense, the idea is you deliver a DNA to the cell. And our idea was we could maybe get particles to go into the cell and design them so once they're inside the cell, they could intercept mRNA. It's the message, right? The mRNA is bound, and when that happens, it is selectively degraded because it's in duplex form, can't get to the ribosome. We stop translation and the production of a protein associated with a given disease. So this is a way of correcting disease cells. This works. There are polymeric materials that allow you to take nucleic acids in, and you can do this type of regulation. The problem is that it doesn't work well. From a therapeutic standpoint, you almost create more problems than you started with by using a lot of the polymeric materials that are out there. Big problems in terms of delivery, how you get them to the right cells. The stability of the nucleic acids. There's also something called siRNA pathways. When you go to RNA, those are horribly unstable. They get degraded very rapidly by nucleases that are present. Targeting, intracellular localization. If they go in by endocytosis, how do you get the active agent out of the endosome? And then you have to have a high efficiency, and this is really critical, low toxicity. And that is a really tall order because, as I said, not only are the nucleic acids, foreign ones, naturally toxic to some extent, the polymeric materials typically used to deliver them are oftentimes worse. So, people have tried lots of different ways of getting nucleic acids into cells with varying levels of success. The ones that really matter are probably these two, but really I'd say lipoplexes in particular. Polymeric structures designed to be positively charged in complex nucleic acids and effectively trick the cell into picking up the nucleic acid. And so that became kind of our benchmark. The question was, would we have a system, if we could do this, that might have advantages over the polymeric structures? Why might we think there'd be an advantage? Well, our initial naive hypothesis was if we could get the particles in, we know they bind better than free nucleic acid, so they'd be better sponges of mRNA once inside the cell, and therefore perhaps more effective agents. The reason I say it was naive is I proposed this to a study section, I'm sure yours here or mine back in my home country, it would have probably been rejected summarily without even being reviewed. They'd say, Mirkin, this is great, but you should spend more time on materials. You don't know what you're talking about on gene regulation. Because everybody knows that to get nucleic acids into cells, you have to complex them with positively charged materials. Calcium, for example, polyplexes, lipoplexes, anything you'd like. And so everybody goes down this particular path. And what I'm talking about is delivering these particles without a polymeric agent. They're some of the most negatively charged entities you can make because they've got a shell of DNA, and we're hoping that they're going to get across the cell membrane, and once they're inside the cell, they're going to be better agents. And that's the real naive part of this. Well, sometimes it's worth doing what I call Friday afternoon experiments. Because as I tell the students, if it doesn't work, just don't tell me. If it does work, we'll celebrate, and we'll keep going down that particular path. And so this one worked, and we started celebrating it immediately because they said, you got to see this again. I put these particles in a cell culture. These are the cells. They're almost black in color. They've got millions of particles per cell. They are going in like gangbusters. This is no extra material, no electroporation, no polymeric material to help them go. They're naturally picked up by the cells. They're some of the most universal agents in terms of cellular uptake. This is not just a nano effect. In other words, I can take those same particles and coat them with BSA, bovine serum albumin, and the cells just stare at them. So there's something very special about that oligonucleotide shell, that spherical structure, that leads to this type of uptake. Well, I'd give these talks, and they'd say, oh, that's nice, Chad, but you picked the easiest cell line. You pick my cell line, it's not going to work. We've gone through now over 50 different cell lines. We've only found one cell line that doesn't pick them up, and those are mature red blood cells. And the reason is they don't endocytose. Every one of these cell types, primary cells, tumor models, will pick them up. As I'm going to show you, tissues pick them up very, very rapidly. It's a big issue in terms of working with them, in terms of getting them on your skin. You have to be aware that they go actually right through skin. Very, very exciting in that regard because this is one of the biggest challenges in terms of this whole area, getting them in and getting them in efficiently and not having to carry an extra material along the way. How are they getting in? That's the science in this, right? So we began to study this, and so this is the lore. This is why you would think they wouldn't go in. You've got this negatively charged membrane, negatively charged particle. What is going to be the mechanism to move them in? Well, we didn't know. We began to study this, so we said, well, maybe they're going to be better if we decrease the coverage of oligonucleotides. And so we could make particles where we would dope in polyethylene glycol, and by controlling that ratio to the ratio of polyethylene glycol with an alkyl thiol N group to the oligonucleotide with a thiol, we could control the amount of oligonucleotide in the surface of the particle. And you can do that. And in fact, you can see here's the surface coverage. It's the oligonucleotides per particle going from 0 to 80. So this would be purely pegylated, and this is a pure oligonucleotide in this particular case. These are smaller particles than the ones I showed you before. And this is the interesting result. You look at uptake, it's counterintuitive. The higher the oligonucleotide coverage, the denser, more densely packed, the greater the uptake. And it's significant. So by going to this very densely packed structure, you end up getting a system that naturally is taken up by cells. Why is that? Well, you begin to study this, and what you find is that the particles, when you have this scaffold, can pick up a shell of scavenger proteins. And those scavenger proteins are what's responsible for triggering endocytosis. And in fact, you can see as I increase the number of strands of DNA, I increase the number of proteins absorbed onto the surface of the particle. So it's actually this complex that's being recognized by the cell, which triggers endocytosis and gives us this great uptake. So we're kind of hijacking the natural cellular machinery to affect uptake of this very polyanionic type of particle. And I would argue now is probably the right type of design to make these types of structures and to get to systems that exhibit these types of properties. High uptake ability, I'm not going to go through this, but no apparent toxicity. They avoid the immune response. One of the reasons they do that is this shell of oligonucleotides deactivates a lot of enzymes, one of them being the enzymes that trigger the immune response. And there's a very high, you can kind of think of it as denaturing a lot of enzymes that get within that particular shell. Same thing with mini nucleases. So there's a much higher intracellular stability. And the other thing you have with this is the ability to put lots of different types of molecules. I haven't shown you, but we can put siRNA on there. We can put all sorts of intracellular probes. And we can create ways of doing simultaneous gene regulation and detection from this one particle construct. So here's an example. So one of the things we wanted to learn how to do was to take these particles, and I said in the cartoon, the particles are going to go in, they're going to bind to mRNA. And then we're going to look at knockdown. And in fact, the way a biologist does it, they measure the amount of protein that's produced, and they quantify the degree of knockdown based on that, or they quantify the mRNA transcripts by using real-time PCR types of techniques. What I wanted to do was to create a way of using this, but to actually visualize the microRNA or the mRNA binding. And so we came up with a new architecture we call a nanoflare. So what we said was maybe what we could do is take a short oligonucleotide with a fluorophore and hybridize it at the base of the particle. And so this has a sticky end on it so that once it enters a cell, that sticky end can bind to a microRNA or an mRNA target, and it's designed so that the mRNA will overlap the region occupied by the flare, so thermodynamically this is a longer track, it's favored, and a flare is released. Why do I call it a flare? Well, when the fluorophore is bound to the particle, the gold's a great quencher, so that's spectroscopically silent. So as these events take place, we release a fluorophore, which turns on, and now I should in fact be able to image and even quantify this type of binding. And so here you can see the first examples of live cell imaging experiments where you can not only image but quantify the degree of mRNA binding inside the cells. And we're looking actually at a gene called survivin, which is associated with cancer. So why might you want to do this? Well, now you could think about feeding a large cell population these types of particles, and if ones overexpress a certain gene, they're going to light up if you've designed the flare properly, and using flow cytometry, you in principle could sort and differentiate these. And in fact, that's exactly what you can do. And so here you can see, using a flow cytometer, we can take cells, fire thousands of them through, and separate the ones that have the genetic cargo from the ones that don't, or above a certain level from the ones that are below a certain level. The only other way we could think about doing this, some of you might be familiar for the aficionados, is to use another known chemical construct. This would be the molecular approach, called a molecular beacon that has a fluorophore and quencher here. And this is designed so that when it goes inside a cell, it could bind to the RNA, and that would push the fluorophore and quencher apart. You'd have to carry that in with a commercial delivery agent, which we've done. The problem with this is, this doesn't have that structure which resists nuclease degradation. So this gets degraded so rapidly in the cell, you get so much background, you can't differentiate the two types of populations. And so it's all of these composite properties that leads to this type of capability. Okay, what about the real deal? Can we make a difference in terms of developing therapeutics? And we're not there yet, but we're pretty close. And these are some very recent, very exciting results, none of which have been published, that show in a glioblastoma model, that's brain cancer, very deadly disease. You get glioblastoma, typically you have 11 to 13 months to live. That's the average life expectancy. It's one of the worst diseases out there. One of the problems is, how do you get therapeutics there? How do you get therapeutics inside, in this case, cells? And how do you flip the right types of genetic switches to cause those cells to selectively die and not kill the healthy cells? This is the problem. These tumors are very aggressive, and they're also very resistant to many of the conventional ways of delivering chemotherapeutics and also genetic-based materials to them. And so here you can even see, even with molecular dyes, they won't penetrate. These are called neurospheres. They won't penetrate them. But with these types of particles, they not only go in, they go all the way to the core of the tumor. And we see that over and over and over again. I told you skin, you can put it on ovaries. So we've done with reproductive biologists experiments there. And in this case, one of the toughest models, glioblastoma type models. So this is done actually with a guy named Alex Stegg, who's in the medical school at Northwestern. And he has a target. Stuart Schreiber was talking about one component of this family, BCL2L12, which turns out to be a very nice target to affect the way the cell avoids apoptosis. So these cancer cells are immortal. They avoid cell death. But the idea is that if you target this, you can get a selective cell death in those cells that take them up. You have no effect in the healthier cells because this is overexpressed in the cancer cells. And it also sensitizes animals ultimately to treatment with chemotherapeutics. To make a long story short, these particles go into these cells extremely well, and you can get very effective knockdown across many different types of tumor types. Another guy at Northwestern, a guy named Tom Mead, is an expert in imaging. And so we thought what we'd like to be able to do is actually image this not only in cells, but ultimately in vivo. And to do that, what you can do is take this construct and now add another component. And this shows you kind of the versatility of it. You can synthesize oligonucleotides with azide-terminated groups. And you guys see azides, any chemist immediately thinks of click reaction. You can now click on gadolinium agents and begin to create a sea of gadolinium on these oligonucleotide constructs. And so now when these go into cells, you get phenomenal contrast agents. What that allows you to do then is to look at animals where you, for example, ask the question, if I take and put some of these particles, let's say by point injection in the brain, how far do they penetrate the brain? How deeply do they get into tumors? Well, here are images, MRI contrast agent images, where you can see the white areas are clearly where we're getting particles diffusing from our point. So we just took a little needle and we put them in a highly concentrated form, and then we're watching their diffusion, their ability to penetrate cells. That's kind of nice. The better way to look at this, though, is through 3D imaging. And so we have phenomenal imaging capabilities at Northwestern. And so this is actually looking at the brain of a mouse where we've put a point injection of the particles. And usually, those, if you delivered nucleic acids without particles, they'd be localized in a very tight spot. Here you can see the result of their movement. They penetrate roughly 20% of the brain after 96 hours, which he had never seen before. And so what you have here is not only an agent that's very effective in terms of knockdown, but one that has this ability to move through tissues very difficult to transport through, or tissues that are oftentimes very difficult to transport through, and get very effective knockdown. So then we said, okay, what happens if you take animals and you inject them systemically? How do we get them in the brain? Originally, we were thinking, well, we'd have to do this post-surgery. We'd have to deliver the particles and get local delivery. That's why I did that point injection. But this is an incredible result. We've done these early experiments where we've injected mice, tail vein injection. The gold is fantastic because we can use ICP mass spec to map where it goes. We have very little gold background in us, and mice have very little gold background in them. So I can quantify and do a full mass balance of where the gold goes. And in this case, 1% gets across the blood-brain barrier. And that sounds like a small number. That turns out to be an enormous number. These are big, big constructs, and you would expect actually 0. They get across the blood-brain barrier, they accumulate in the brain, and here's the beautiful thing, they also accumulate in the tumor. And in fact, the tumor's a little more leaky than the rest of the brain, and so you get a higher concentration of them in there. And then what we looked at, and we just duplicated, we started out with a model of 6 mice, and we've now gone up to a larger cohort, I think it's 16, that we just duplicated literally last week. And the same result we got. So we're pretty sure this is right now. When we target this nucleic acid construct, or target the BCL12 construct, what we end up getting is increased animal survival by about 25 to 30%. So what we know is that the particles are getting to the brain, localizing in there, and then we can take the tumor and look at whether or not we have apoptosis and also stop cell proliferation. And in both cases, we know we not only get gold accumulating there, but we're getting knockdown in there, in live animals. And so this is extremely promising because what it means is that you have now a whole series of new candidates you can think about in terms of developing new types of therapies for one of the, I think, the worst diseases out there. And the beauty of using this is the bar in terms of getting something that's through, for example, the FDA is much lower because there is such a need to develop these types of therapeutics. Okay. So these are kind of the take-home messages in terms of what I'm trying to get across here. I want you to think about these types of constructs as completely new chemical constructs. Now, if you're looking at this and watching it carefully and critically, I'm sure these two guys are, you say, wait, you told me that you've got a spherical nucleic acid, but you really have a carrier. You've got a gold particle in there. And that's kind of like the polymer that carries in the nucleic acids via the other route. And so, we began to think about that and said, well, our hypothesis is that this dense shell of DNA is really quite critical in terms of transfection. So we wanted to come up with a way of making a structure that's pure spherical nucleic acid construct. And very recently, to show you the last 2 slides, we can make oligonucleotides that have propargyl groups that can bind to the surface of the gold particle, and the gold can act as a catalyst to cross-link these structures. So now what I have is a cross-linked shell of a oligonucleotide in the surface of the particle, and I can then add iodine and dissolve, or cyanide, and dissolve the gold, and now I'm left with truly, as close as I'm going to get to it, organic spherical nucleic acid. And I can ask the question, will those go in? So this just shows you can make these structures. You can, as you imagine, change the size and shape of the template. This is before treatment, after we treat and then begin to dissolve, and then you can see the spherical nucleic acids left behind when we stain them, and you can just watch the color disappear. The particles are trapped, so you never see them aggregate. They just slowly dissolve until you have all gold salt in solution. But here's the kind of nail in the coffin type of experiment that tells you this architecture is truly special. Remember, this was the key Friday afternoon experiment. This is that same experiment with pure spherical nucleic acid. We put a fluorophore label so you can track it. It goes in like gangbusters. In fact, we've quantified it. It's almost identical. So, what I want you to do is to think about these types of structures as fundamentally new forms of nucleic acids. They are very similar in terms of components, very different in terms of structure, and clearly very different in terms of properties. And I think they have a pretty big future in many of these different types of areas, and we, among others, are going to begin to, I think, try to exploit that and figure out how we can really use them to maybe develop some tools that can have a big impact in terms of treating some of the worst diseases out there. Okay, let me finish by acknowledging the folks that have done the work. I always do that. I always get that reaction. This is not my group. No, I took this all the way. I'm just kidding. This is my group, but it's my group over many years. So we just had a 20-year reunion and brought everybody together. We have a very large group, but this is ridiculously large. But many of them, because I talked about 15 years, contributed to the many different aspects of what I've talked about. And it's really, as I said, many people coming. We talked about interdisciplinary, from different fields, different ideas, different countries, providing input that have really driven the development of this project. And the other thing I have is I've got great collaborators. I mentioned Alex Stegg. He's on the brain side. Amy Pollard, I took out all the skin cancer work. These are fantastic for delivery through the skin. Shad Thaxton, who's now a professor at Northwestern. And George Schatz, who I should mention, he's done really all the theoretical modeling of many of these types of structures. And he helps provide a kind of a sounding board in Computero that allows us to challenge a lot of our experimental models. And then funding was provided by these folks. And I will stop there, and it's party time. Thanks.